Hello, my name is Rita Hayford from University of Delaware, and the title of my presentation is Building a Text Mining Pipeline to Retrieve Literature to Study Stress Response in Arabidopsis. We have presented some of the key points and figures of our poster presentation and presented them on these slides. On this slide, you see Arabidopsis plant, an example of environmental stresses that can affect this plant. And so, for example, you see an example of biotic stress, which is botrytis, an example of abiotic stress, which is drought. Arabidopsis is a model for plant research, and it has widely been used as a reference for stress response study in plants. In this study, we focus on Arabidopsis. However, we would like to expand to other plants in the future. One of the challenges in plant research is how they adapt to climate change. Our goal is to link stress-responsive genes to their function. Literature on plant stress is increasing, and genes that respond to environmental stress in plants have been reported in the scientific literature, and this keeps increasing as well. And this is indicated in the figure as shown on the left side of the slide. And below the figure, we have shown a script that we use to query PubMed for this information. And so our objective is to establish a text mining pipeline to efficiently retrieve publications relevant to Arabidopsis genes and stress. We would also like to connect resources to understand plant stress. The outcome of our study could assist biologists in plant stress tolerance study. This will also complement existing databases with knowledge about plant stress genes. And this slide shows a workflow to retrieve genes and link them to their function in Arabidopsis. For the initial study, we evaluated both in-house and publicly available text mining tools, which include Testpresso, Pigeon, PubData, and EuroPMC. Testpresso is a text mining system that allows for literature search using keywords and control vocabulary terms. Pigeon identifies gene names and link them to a database entry. PubData also helps to retrieve bioconcept annotations. We use Pigeon and PubData complementary to each other because Pigeon has high precision and PubData has high recall. We obtain functional annotation sentences as well as Go terms from EuroPMC. We selected some keywords and used them as inputs for the literature search from the Arabidopsis corpus in Testpresso. The number of abstracts obtained from Testpresso were passed to Pigeon and PubData to retrieve gene mentions. Pigeon and PubData were run parallel to each other, and then to EuroPMC to obtain functional annotation sentences for some of the abstracts. We plan to store the data that we have collected in MongoDB. In the future, we would like to disseminate the outputs of our results in the iTestMine framework. iTestMine is one of the in-house text mining tools developed at University of Delaware, and it integrates text mining tools and extraction relation results to obtain a comprehensive knowledge of scientific literature. Here we have presented some of the preliminary results that we have been able to collect so far. And in this summary statistics, you see data collected on the number of abstracts retrieved from Testpresso, the number of gene mentions collected from Pigeon and PubData, as well as functional sentences and Go terms collected from EuroPMC. And usually when we use publicly available tools, there could be some challenges. And one of the challenges that we encountered has to do with gene reef limitation in providing all the functional sentences. And so for the abstracts with no functional sentences, we collected information on the stress genes and their relation to the Go term. The Venn diagrams below the summary statistics indicate the unique and overlapping abstracts as well as gene mentions retrieved from Pigeon and PubData. On this slide, we show a section of the data that we have curated over the time period. Here, we wanted to bring out some terms that researchers could review for their purposes. In addition, we have been able to submit new citations from the curated data to the UniProt database. Interestingly, we were able to classify some of the data collected as high confidence. And this is when you have a gene mention, functional annotation, and goal term identified in the same sentence. Here on this figure, we have captured in iTestMine, which I mentioned before, MYB2 gene, which we collected as part of the stress genes. The figure shows MYB2 regulation of microRNA involved in abscisic acid response. Our work could provide a link to the underlying mechanisms. And so through an experiment, MYB2 is upregulated in stress. And by using iTestMine, we could identify the underlying mechanism. In summary, we showed integration of existing text mining methods to effectively retrieve and enrich bibliome content related to stress response in Arabidopsis. The information we have collected will be stored in a database that interprets with iTestMine to highlight other additional relevant information or relations. The methodology we have applied here will be extended to all plants. We do welcome any suggestions that will be useful to our study as we continue to enrich this data. With this, I would like to thank you for listening.